Hello. Welcome, everyone. It's great to see everyone both in person as well as over Zoom right now for the third of the four Wednesday evening at the Genome seminars hosted by the Department of Genome Sciences here at the University of Washington. Thanks for everyone who's showing up for this. So I'm really excited to actually announce for our speaker today, Gaurav Bhardwaj, who's one of the associate or assistant professors in the Institute for Protein Design, as well as the Department of Medicinal Chemistry here at the University of Washington. So as background, he did his training at the Penn State University and then came out here working with David Baker for his postdoc, really did some exceptionally innovative stuff using AI to leverage that for doing protein design work, which I'm really excited to hear about. Had numerous phenomenal papers as a result of this, very well recognized in the field as a result of this. And you really, I think, cutting edge in terms of how we can push the boundaries on not just doing cool stuff from the AI perspective, but actually making it in terms of clinical useful diagnostics. And that's actually part of, I think you're co-founder of a company as well, doing exactly that. So it's really lovely to welcome you here. And I'm really excited to hear more about your research today. So thank you.
Thank you. And thank you so much for the invite and opportunity to join this meeting. And I'm really excited about this. So my lab actually works on designing peptides, designing them on a computer first and then making them in the lab. How's the sound in the back? Is the sound good? Okay, awesome. So the reason we kind of started working on this, and like a lot of other people in this space, we believe that peptide drugs can fill the gap between small molecules and large molecules, right? And I laugh about this because this is a slide that pretty much every peptide chemist has as their first slide. And the idea is that, look, small molecule drugs like aspirin are really stable, but they tend to have a lot of off-target effects because they bind to a lot of other things, right? Large antibody-based drugs kind of solve this problem. They're very selective, but they're large proteins. They're not very stable. They require cold chain. They cannot get inside the cells. So pretty much every peptide chemist thinks that hopefully we can make peptides that can be as stable as small molecules. You can put them on your shelf for years and nothing bad happens, and make them as selective as antibody-based drugs. And of course, we are not the first people to think of peptides as drugs. A lot of people have tried this. People are trying this. But one way people have tried to do this is look towards nature. And there is this amazing, fascinating field where people have found really useful drugs from cone snail venoms. There is a very potent anti-pain medication that actually came from a cone snail venom. There are sort of like a lot of these like plant cyclic peptides that are being used as bee-safe insecticides in Australia. So there are a lot of these really cool stuff. There are some really useful peptides from scorpion venoms. There are a lot of these knotins, like knot-like peptides, that are being used or being tried for cancer targeting. So nature is great. And in fact, I'm not going to run this video today, but I am going to highlight, definitely go back today, search for cone snails hunting for fishes on YouTube. There are these amazing videos where these very tiny cone snails, when they actually hunt for fishes, they inject hundreds of different peptides into the fish. And some of those peptides have become very useful drugs because cone snails do not want the fish to feel pain when they actually sting them. And that's how we found a very potent anti-pain medication. Right? They are sort of like these versions of like very fast-acting insulins that have been found from cone snail venom because cone snails want the fish to go into hypoglycemic shock. So there's this really, really cool, there's a whole field of these people looking through venoms of like scorpions, cone snails, spiders, and are finding these very useful peptides, right? But I always joke about, right? Like it's very fun field. It's great to go find something in nature, but you can't walk up to a cone snail and ask for it a new drug when you need it, right? Cone snails don't listen to you. So other thing that people have tried is this idea that what if we make all possible combinations of peptides, right? We can't ask the cone snail for a new drug. What if we make all possible combinations of peptide sequences or amino acid sequences, throw them at our target protein, and hope that we can find the function that we are looking for, right? So people have tried doing sort of like displaying many random peptides on the yeast. People have tried to display peptides on the fish, or even sort of these chemically synthesized libraries, right? In principle, it's great. But generally, if you start thinking about the possibilities or the number of combinations is so huge, right? Even with natural 20 amino acids, if you make like a 10 amino acid peptide, it's 20 times 20 times 20 times 20 times 20, right? If we are chemically synthesizing, we can use hundreds of non-canonical amino acids. So then the number of amino acid possibilities are 100 times 100 times 100 times 100. The space explodes so quickly. So none of the library methods, even the best library methods that we have, are really just subsampling all possible combinations. So if we are making a random library or subpopulation of a random library, we are really hoping that the function that we need exists in that library. Right? So sometimes it works. I used to be in a lab that actually used to make these libraries before coming to David Baker's group. And sometimes you do find that function, but most of the times you're really just hoping and either you have a very weak function or you don't find the functional peptides at all. Right? So when I joined David's lab, the idea was that what if we could computationally design peptides, and that would, in principle, complement both these approaches. Right? In principle, we can design peptides that we have not found in nature, or nature is not even sampling them. What if you could design peptides beyond nature? Or instead of making completely random libraries, what if we design first and then make very focused peptide libraries against those targets? So that was sort of like our goal behind why do we want to do computational peptide design, right? So I joined David's lab, and one of the first methods, and this is pre-AI days, right? We did not have any of the AI tools at that point. I was a postdoc in that lab. Everyone else was designing big proteins. And I realized pretty early on that most of the methods that work for designing large proteins do not work for small peptides. One of the things that people were using was break down all protein structures in PDB, break them down into smaller fragments, and assemble new proteins from these fragments. If we are trying to design peptides with non-canonical amino acids, there are no fragments in the PDB with non-canonical amino acids. So how do you assemble new peptides if you don't have those fragments? So we actually had to borrow this method called kinematic closure from the field of robotics. And at the face of it, it's very, very simple method, right? Really what we are doing is you start with an elongated peptide chain, it's just backbone. And if you start twisting any of these backbone bonds, right, you're twisting them in space, every time you twist it, it changes its shape, right? If you just do it very fast enough, you just keep some random perturbations, you twist this chain, it takes some shape, you twist some other atoms, it takes a different shape. You really are just doing this to find a conformation that would give you a cyclic peptide backbone. So generally, this is one bond at a time and just rotating that in the space, right? And if you do it randomly, you can find conformations that would form a cyclic peptide backbone. There are some sort of like optimizations built in, but conceptually, that's the idea. And once you find a cyclic peptide backbone, then you can design an amino acid sequence that would try to minimize the energy of the system. So the whole goal was try to find peptide conformations that would fold into low energy states. Right? And this is physics-based stuff, so no AI. Everything is driven by trying to get to the lowest energy state because that's what things fold into. And because it's random perturbations, every time we run this, we keep sampling different shapes of peptides. Right? So this was the idea. And essentially, when I say we borrowed it from the field of robotics, it's very similar to how we decide to move robotic arms. Right? If we have to reach a certain point, we have to decide how to twist the arm of this robot, how to rotate these limbs. Now think of exactly the peptide. Instead of trying to reach somewhere, you're trying to reach back on itself. That's how you're sampling cyclic peptides. Right? So we did this. And what this allowed us to do was design peptides with shapes like this. This peptide, half of this is made up of natural amino acid. Half of this is made of the mirror amino acids. And just to make the point, this is the kind of shape that nature is not evolving in. So we are able to, at least on a computer, we are able to design shapes that nature doesn't evolve in. Right? Some of, again, like other shapes that have like some canonical amino acids, some unnatural amino acids in there. But this is just on a computer at this point. Right? You can make up anything on a computer. The real test is, does it actually work if we go to the lab and make these peptides? And of course, like, I wouldn't really be talking about this if it would not have worked, right? Um, so we go to the lab, we make these peptides, solve their structure by NMR, and when we superimpose the structure of the peptide, which is in gray, to the design model on the computer, they match up almost exactly, right? Down to sort of like very close to 1 angstrom matches between the design model and what the structure on the real structure by NMR looks like. Okay? Uh, and including things which are like half made of canonical, half made of non-canonical amino acids. So we are able to sample many different shapes. We go to the lab, make them, they do fold into the design structure. Right? We have also, the other part of this is I was talking about the stability part of it, right? You can take some of these small, compact, constrained peptides, heat them up. So this is a CD spectra. You can heat them up to 95 degrees Celsius, and they still retain their shapes. You can actually put them into 6 molar guanidine hydrochloride, they still retain their shapes. And this is not specific for design peptides. Even the natural knotted peptides tend to be highly stable. And this goes back to this idea that now we can actually keep them at room temperature for a long time. We can ship them at a room temperature at a long time. They retain their function. So we don't require cold chain. We don't require sort of like any special handling around these peptides. And that's an advantage for these small compact peptides compared to antibody-based drugs. The next thing that we did is we extended the same idea of kinematic sampling to design even smaller peptides, anywhere from sort of like 7 amino acid to 14 amino acid. And the thinking behind this was these small peptides tend to have more drug-like properties. We can convert them to drugs. There was a lot of interest in the field around them. But the belief, at least even when I started working on this project, was that most likely it would be very difficult to design peptides of this size that have a single low energy structure or a one conformation. Assumption was most of these peptides, because they have no secondary structure, would most likely be flexible and have multiple conformations. We were surprised that you can actually find low energy minima even in these small peptides. And when you solve the NMR ensemble, it actually matches again very closely for most of them, very, very closely to what the design model on the computer looked like. Right? So at this point, sort of like I was completing my postdoc, we published all this work. The next step for us was like, look, we can design peptides to have the shapes that we want them to have. We have a very precise control over the structure of peptides. Up till this point, we were not designing for any function. We were just designing to have, shape number 1, shape number 2, just many different shapes of peptides, right? There's no function at that point in our mind. Then we started thinking was like, look, if we have precise control over the structure, what do we want to do with it? And by the central dogma, if you kind of like can control the sequence, you can control the structure. If you control the structure, you should be able to control the function, right? So at this point, we were pretty excited because we thought if we can design peptides with angstrom level accuracy, we can go after any function we want, right? And we have few things in mind. Some were, what if we design peptides to bind to therapeutic targets? Most of the times, drug discovery involves, given this target in gray, can you design a peptide or a protein that binds to it? So that's one function or class of functions we wanted to go after. And I'm going to come back to it. Or design peptides that would self-assemble to bind to metal ions or do some catalytic activity. We are working on this now, but I'm not going to spend too much time on this today. And the last part was, can we design peptides that would get across the membranes? And the reason to do that, and I'll come to it. So I'm going to focus on this part first and then go to this. And the reason we wanted to design peptides that get across the membranes is a few different applications that we have in our mind. Right? One is, a lot of drug targets, a lot of good drug targets happen to be inside the cell. Right now, most of the antibodies are not getting to those targets. Right? Most of the small molecules are not good at blocking large protein-protein interfaces that are happening inside the cell. So if we can get inside the cell, we should be able to go after these targets that are traditionally considered undruggable. The same idea, if you want to make peptides to be orally bioavailable, most peptide-based drugs right now are injectables. And that's because they cannot cross this epithelial gut barrier. So if you want to get across this epithelial gut barrier, one way to do this is to make a peptide that can passively float across this whole cell. So from our gut, get across the epithelial cells. And the same exact idea is if you want to get a peptide to the brain, we should be able to cross, or one way to cross blood-brain barrier is to make a peptide that can get across this endothelial cells. Right? All of these, at least one core principle here is passive permeability. So can we design peptides that get across these different lipid barriers or lipid-based membranes in the cells of different types? Okay. So when we started on this was an interesting problem. We had no clear guidance on what makes a peptide membrane permeable. In fact, when I started on this problem, there was a lot of ideas in the field. But for every idea that said, oh, having this feature makes a peptide membrane permeable, I could also find a paper that said it doesn't do it. And it was true for almost everything. There was a complete chaos in the field in terms of like, somebody would say having 4 arginines in the peptide makes it permeable. And then there would be another paper that says, no, that's not going to work at all. Or having more D amino acid makes it permeable, which made more sense, or having cis peptide bond makes it permeable, right? And there were a few examples from nature that people were pointing to that look, cyclosporin is permeable. Can you make something like that? Right? So we said, look, we don't know what makes a peptide permeable. We know how to make peptides into different shapes. Right? We'll take every idea or some of the ideas and design hundreds of peptides that satisfy idea 1. Just different shapes that satisfy idea 1 or idea 2 or idea 3. Right? We'll test them in the lab. And at least at the end of this, we will understand what is a designable principle for us, at least, to make it membrane permeable. So one of the first things that we tried was to make hydrophobic peptides that have no hydrogen bond donor or no unsatisfied hydrogen bond donor. And the idea behind this is if you have unsatisfied hydrogen bond donors like this, then they are going to be hydrogen bonding to water. And when you're trying to cross the membrane, it's very hard to take a water molecule across a lipid membrane. So if you've done those experiments of trying to mix lipids with water, they don't. So if you're trying to take water molecules and drag them across the lipid membrane, it doesn't work out. There's a huge energetic penalty for it. So what we could do is we can design peptides that are very compact. Every hydrogen bond donor is already satisfied by a carbonyl from another amino acid. And if there are some positions that are left, then we can convert them into this N-methylated amino acids. These are non-canonical amino acids that we can buy, we can chemically synthesize. Right? So, and that's one way to mask any exposed unsatisfied hydrogen bond donor. The other idea was, what if we make peptides that don't have just one single low energy conformations, but can switch between their conformations? So there would be one polar conformation outside the lipid membrane, but as they're going through the membrane, they can change their shape and now hide all their polar groups so that they are favorable in the lipid as well. But then on the other side, they can switch back again. So instead of a peptide with one single low energy conformation, we can switch between conformations. So we designed sort of like hundreds of peptides to satisfy these ideas. And then when we test them in the lab, we realized that majority of them actually are membrane permeable. And this is sort of like an in vitro assay, is a common first experiment in this field, where you literally have lipid membrane in the middle. You put peptide on one side of this trans well, measure how much peptide crosses in a given period of time. Right? And generally a value around here, like 1 into 10 to the minus 6, like right around here would be considered good permeability by pharma standards. So we had 6 amino acid to 12 amino acid peptides that are considered too big to be permeable, all designed to satisfy one of those 2 criterias, and they're all showing some really, really good permeability values. Next, we tested them in KCO2 assays, similar idea, but now we actually have gut epithelial cells growing forming a perfect monolayer. And then we measure how much peptide crosses. This time, not just crosses the cell, but also crosses against the efflux transport. So our gut epithelial cells have efflux transporters. Their job is to throw things back out. So if we want to cross the gut barrier, then we have to overcome the efflux transport. So in this case, that's what we are measuring. And the same idea that we actually had a lot of peptides, different shapes of peptides that we are actually able to get across these oral bioavailability in vitro assays. Right? Another thing for us is like, sure, they're permeable, but are they crossing in the design state? Otherwise we are just getting lucky here, right? So then we solve the structures of all our design peptides. So it's sort of like, around 25 to 35 of them. And I'm not showing the side chains of those peptides just for clarity, but they're there. And now the design model is in blue, and the structure, the crystal structure that we solved is in orange. And some of these were like 0.2 angstrom matches between the design model on the computer and the experimental structure that we got. We have tested some of these peptides in animal models, and we are getting really good oral bioavailability values. One of the 11-mer peptides had an oral bioavailability of 41% without any special formulation. So this is like feeding the peptide in no special formulation. It survives all the proteases, the harsh environment of the gut, and then gets absorbed and makes it to the systemic circulation unmodified 41%, which is truly sort of like a remarkable number to get to. Because most peptide, even a lot of formulation strategies that people use now, get 1 to 2% oral bioavailability. So we were, this was sort of like, one of the big challenges in the field, and we were very happy that we made a lot of progress on this thing. But the next thing that we tried to do is make binders to targets, because if you're trying to make new therapeutics, that's the big thing, right? Somebody comes to you with a drug target. The goal is to design a peptide that perfectly fits into this pocket, right? And we started with our physics-based methods. The same idea, this like kinematic sampling, twist, 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 find cyclic peptide backbones, design an amino acid sequence. But this time around, do the same thing, not outside, but just directly into a target. So imagine this is the pocket you're trying to target. You start somewhere, sample a lot of peptide shapes, design amino acid to fill this shape, right? And we tried this on multiple targets. And every single time, we got the same frustrating result, which is we kept getting these low micromolar binders. So we have done this against like the SARS-CoV-2 main protease, some of the antibiotic targets, every single time low micromolar binders. And there are people in the field who can make billions of random peptide libraries, a peptide library with billions of members and get nanomolar binders. Not all the time, but definitely enough number of times. So that's kind of like the benchmark, right? To get to sort of like a nanomolar binder as quickly as possible. Now, we kept getting these micromolar binders. And the problem was, on a computer, we can sample millions of or billions of combinations, right? We are sampling massive numbers. But because in the lab, we were limited to testing 10 to 20 peptides, we have to go from these millions of design candidates on the computer to selecting 10 peptides or 20 peptides that we are going to make and test against a target. Now, is this a problem that we are not sampling good binders, or are we not selecting them in our top 10? And I think as of today that it is still a ranking problem, that we do sample high affinity binders, we just never pick them in our top 10 or top 20, right? But we tried to kind of improve our ranking schemes and few different things. But this was not like, these were like 2.5 years of very frustrating lab experience where we kept going after targets, trying to fix things. We were not breaking the micromolar barrier that we were saying, right? At the same time, the field of protein design was getting all these like new AI methods. AlphaFold 2 had come out, AlphaFold 1 had come out, Rosetta Fold from David's lab had come out. And in the protein design space, people were making amazing progress. Right? The accuracy of protein design was improving, the success rates were going up, they were able to design peptides or proteins very fast. And we have used those methods. So one of the methods that came out of David Baker's group is this RF diffusion. We didn't contribute to this method, but we've used this method to design protein binders to different targets, right? And generally how it works, and I'll come back to this idea, is that given a target, which is in gray here, you start with a randomly thrown amino acid at this target, and then you stepwise or iteratively denoise it to form a protein binder to it. And then you design an amino acid sequence. Then you filter your designs by AlphaFold 2 or AlphaFold 3 or any of these like new structure prediction methods. We have used this method on multiple targets. This is a virulence factor from a bacterial pathogen that is highly infectious. Right? So we are trying to design an inhibitor of this virulence factor. We tested 15,000 designs in this target, but we get a picomolar binder. Another target, another sort of like a surface protein from a bacteria, one of the escape pathogens. In this case, we only tested 96 designs, and we got a picomolar binder. So in the protein design space, things were moving very fast, right? We are able to use these methods, get picomolar binders very fast by testing only 96 designs. As a peptide lab, the question was like, how much of these methods or similar methods can we build for peptide design? Because I would love to make a picomolar binder for a peptide by only testing 10 designs or 20 designs, right? So some of the problems still exist as like, there's not enough training data for peptides. There's not enough training data for non-canonical amino acids. So how do you build a new model from scratch? But we said, look, let's ignore all those things for now. How much of the information in the protein space can we adapt for peptides? Can we even take a protein structure prediction model like AlphaFold, adapt it to work for peptides? It was never trained to do that. By default, it doesn't work with peptides. Can we at least tweak it to work for us? That's the first thing that we did, where we modified AlphaFold to work with cyclic peptides. And just not to design, but to predict the structures. And we made some changes to the code. We can ignore this for now. But ultimately, the idea was once we made these modifications to AlphaFold 2, we were able to predict the structures of peptides. These are the peptides that AlphaFold 2 had not seen during training. And generally, whenever AlphaFold 2 seems pretty confident about the prediction, so this is like a confidence score. And this is how close is it to the real structure. And most of the times, on these structures that AlphaFold had not been trained on for cyclic peptides, if it feels confident, it tends to be very close to the real structure, right? And I'm showing a couple of examples here where the prediction is in kind of like gray and the real structure is in different colors. So AlphaFold 2 seems to be pretty good at predicting the structure of peptides. Now, the fun part is if you can predict something with AI, you can always invert the network to design things with it. Right? You can walk back through the network. And what I mean by that is, let's say you start with a random sequence and do a structure prediction. It's going to look pretty bad, right? Like you pick a random string of amino acids, you run it through this AlphaFold 2 or modified AlphaFold 2. The first prediction is going to be pretty bad. But then what you can do is you can take that bad prediction, back propagate through the network, and update the sequence to try to make it better prediction. And you keep doing this, you keep walking back through the network till you have found a peptide that is very highly confidently predicted. Right? So you take the structure prediction network and you back propagate through it multiple number of times till you get a high confidence prediction or structure out of it. Right? That's basically what is shown here. And we did this back propagation approach to design new peptide structures between 7 to sort of like 14 amino acid. And again, the process is called hallucination because the network is hallucinating new peptide structures. And when we solve their X-ray structures, we make them in the lab, solve their X-ray structures, they do match up very closely, sometimes 0.4 angstroms, 1 angstrom, like really, really closely. Right? And this is very similar to what I showed on like, a couple of slides in the beginning, right? We could do this with physics-based methods. So this is not new, right? The difference is what used to take weeks to get one good structure, now we can do one good structure in less than a minute. I laugh about this. One of my postdoc projects, which actually took like a couple of years, I think I can do this now in a week or less than a week. So even though we are just designing similar peptides just for structures, it's much, much faster and also more accurate. Um, and because it's so fast, we can sample many different shapes of peptides that are highly confidently predicted. Right? And once you collect all possible shapes of peptides that you think are very highly confidently predicted to be on the structures, you can use them to design binders. And the way we do this is, let's say this is our target. We can start with a motif that we think binds to this target. This motif could come from another interaction to another protein. You try to graft it on all possible shapes that you have sampled to be high confidence. You graft it, then design an amino acid sequence to make more interactions, and then use AlphaFold 2 or Rosetta Fold 2 to check, is it actually going to fold into this structure, or is it going to bind to this structure? Right? So you design and then check with another network if it's still predicted to bind to this. So we did this for a target MDM2, tested 14 designs, and this time 7 out of 14 have some activity at 50 micromolar, and the best one has an IC50 of around 340 nanomolar. Right? We solved the structure of the target bound to the peptide, the full complex, and it does match up exactly how the design model looks like. Right? So this was fun for us. At least, we are somewhat below micromolar, still not great. Another problem is this is an easy target. Like MDM2 is an easy target. A lot of people have already made binders to this. Right? Other problem is we are starting with a motif which is borrowed from something else. Ideally, a design method should be able to design a peptide binder completely de novo. Somebody gives you a target and you run your method, you pick your peptide and it works. Right? No starting point required. That's what actually changed in like last couple of year and a half now. This idea of like completely de novo macrocycle design using AI methods for us. Right? So we adapted RF diffusion, that idea I was showing of designing protein binders from David's lab. We modified and fine-tuned that network to design small peptides. And this is a work with David's lab, Frank DeMaio's lab, where we were able to actually start, given target, start with the random placements of atoms or residues, denoise it to make plausible looking peptide backbones, and then design an amino acid sequence, and then pick our best designs. And I'm sure you've heard this or seen this somewhere else. Conceptually, it's very similar to how people generate pictures of cats and dogs on the internet. Right? So you start with a real picture, you add some noise, it becomes a little grainy. You kind of train your network to go from this grainy picture to make a sharp picture again. So you're denoising a grainy picture to a sharp one, right? You add some more noise, learn how to go back, you add some more noise, ultimately getting to a point where you can take a picture that looks like white noise, and you can denoise it back all the way looking, a picture of a cat or a dog or whatever you want. Now, the same exact idea applied to proteins. Proteins are essentially pictures in three-dimensional space. Instead of pixels, you have atoms. So you can do the same thing. You can start with real protein structures, add some noise, the atoms are going to break. You learn how to go back to plausible looking protein backbones. You add some more noise, more noise, more noise. And ultimately, once the network is trained, you can start with a completely random placement of atoms, denoise it, back to make a plausible looking peptide backbone. And then you design an amino acid sequence using a different neural network. Since we kind of like started using it, that's the first time when things just it's complete game changer in the lab, right? One of the targets we applied this against is GABAREM. It's a protein involved in autophagy. We use RF peptides, this new pipeline, only tested 6 designs in the lab, and we get a 6 nanomolar binder. And in this case, we didn't start with any motif. It was purely just we took the structure of the target and designed against that completely de novo. Right? Now, it got even more interesting for one of the targets. This is a surface protein from bacteria A. baumannii. We are trying to design inhibitors of this or binders to this. In this case, when we started, we did not have the structure of the target. Like there was no structure available. Our collaborator, Joseph Mujo's lab, had found this protein. We just had the sequence of this. And if you ask me 2 years back, I would have never tried to design against a sequence, or a predicted structure of a target. We always ask for high resolution crystal structures. That's what we are going to design against. In this case, partially because this was a funded project and we had to go present to the funding organization, we're like, let's just try it, right? We didn't think it was going to work, but we predicted the structure using AlphaFold 2 and Rosetta Fold. So we got this blue predicted structure, designed peptides against a predicted structure, tested only 11 designs in the lab, and we get sort of like a 10 nanomolar binder. And that had not happened to us ever, that we can design against a predicted structure, test only 10 designs, and get it to work to a high affinity. Uh, we have solved the structures of many of these complexes that we have designed in the lab, and they match up very, very closely to what the X-ray structure is. It's kind of like yellow. The design models are in different colors, but they do bind exactly how they were designed to bind, and they adopt the shapes that we designed them to adopt. We are actually able to now, we are testing, initially we started by testing only 10 designs or so. We can actually combine this design method now with high throughput display methods. So instead of testing 10 designs, it would be great to test 15,000 designs. So we are doing this against multiple targets now. And that helps with two things. One, we can get to a hit very quickly, even against very difficult targets. The whole pipeline right now practically takes like 3 to 7 days for us to design things. Experimental screening is anywhere from 2 to 3 weeks. But if we really, really have to do this, I think it can come down to less than a week. And we have some ideas of how to get it down to just a couple of days. From picking a target to getting a hit, like that's the goal now, that finish from picking a new target to get validated hits within 2 or 3 days. I think it can be done. Maybe in like a year, we should be there. The other part of this is all the data that we are generating within. So it's not just getting hits, but all the data that we are getting from like testing 15,000 designs against multiple targets. That's very helpful for us to improve the methods even more. Sometimes when we miss something, when we test 15,000 designs, it tells us, why did you think that this should bind when it doesn't bind? So we actually feed all that information back into our design models so that we can keep improving them now. So it's already 40 minutes, maybe just 2 more minutes, and I'm going to stop after this. All the stuff that I presented today was cyclic peptides. One area that we are applying some of these tools is not just cyclic peptides, but stapled peptides, where the peptide has like some sort of like cross-linker and it can stabilize its shape, right? And one area where this is very helpful, and this is a general area that we are very, very interested in, is this idea of designing new antibiotics from scratch. But this is not purely from scratch. In this case, there is this new natural product, or not new, but like a couple of years now, called darabactin. It binds to the surface protein of a protein from escape pathogens, and it has some antibacterial activity, somewhat promising antibacterial activity. The problem with this is it's a natural product that has very weird cyclization chemistry. It's a little hard to see, but there are 3 amino acids kind of like joined together through the side chains. And to synthesize this, it takes 16 to 20 steps of chemical synthesis or total synthesis scheme. Right? And when synthesis is so difficult, it makes it very hard to make many, many different variations of it, try to make improved versions of this. Right? So when we looked at this, we said, well, what if we just keep the good parts? We can design a new version of this. We will keep the good parts that we know are involved in binding to the target. But the difficult part is really not doing anything, just stabilizing the structure. So what if we replace with something that is very, very easy to staple, will keep the same shape, and will still be able to bind, right? And we did that, and we were able to kind of like design sort of like these stapled peptide analogs that actually do have promising or even better activity than the natural product, and is much, much easier to synthesize. And when we solve the cryo-EM structure of our design stapled analog, it does bind to the same side. So this is one of those areas in antibacterial discovery where we keep finding a lot of these natural products that have some good things to them, like their activity, but sometimes they're not stable their metabolic stability is very bad, they get degraded within like few seconds, or they are very difficult to synthesize, is where the design can help a lot, where we just make versions that keep the good parts and replace the difficult parts or unstable parts with a more stable version. So we are doing this against multiple natural products now and making their versions. So I'm going to stop here. I'm going to skip the last slide. Thank people in my lab. We kind of started right in the beginning of pandemic. So perfect time to start a new lab. My first students joined the first week of lockdowns. Um, so a lot of progress, it's really amazing the progress that, people have made. Steven just graduated recently. He was the first author on, the modifying AlphaFold and RA peptide work that I talked about. And a lot of people are collaborators in David's lab and, IPD and a lot of collaborators all over the world. And thank you so much. Happy to take any questions.
Around with the mic if anyone has any questions. If you're online, if you have any questions, feel free to write them in the chat and I can read them out loud.
Hi, thank you. That was an amazing talk. One question that I actually have is, so when you're creating the ideal peptide to the ideal target that you're looking for, have you ever run into any like happy little accidents where you actually accidentally create or you design based on the ranking of peptides that are maybe within your top 10 that would have other applications that you maybe did not anticipate that might be for like more interesting to investigate?
That's a great question. One of this, a lot of times we're not really looking for other applications, so we may miss them. One area that I've always wanted to go back to is, some of the shapes that we designed early on would aggregate, but then aggregation can be used for some of the applications that we want now. Like is it aggregation or is it assembly into, like some of the beta stranded peptides can definitely assemble. And one of the areas that we are playing around now in is self-assembly of these small peptides, but then the assembled structure may be able to have catalytic activity. So we are definitely looking back at some of the old peptides that did show some aggregation behavior and now trying to predict those sequences in like different assemblies and see if they actually form those assemblies. So that would be one thing that comes to my mind right away. Other than that, we may have been missing like a lot of stuff because we are so focused on designing for this function and test for that function. So yeah.
This protein binders, did you try to fuse these binders to some other protein and then try to see that if they're binding again to the target protein?
Why do you ask?
The binders can be used for a variety of applications, right?
Yeah. So I think in that sense, the part that I actually skipped right now, and maybe, is we are trying to design sort of like peptide-based degraders, where the idea is peptide can go bind, recognize another protein, what we are binding to target, and then we can link it to something that would degrade this other protein. Right. So in fact, like one of the work that we have ongoing with Ning Zheng's lab is in a rare pediatric cancer, this hepatocellular carcinoma, people lose these 400 kilobytes region, which fuses two proteins. And this fusion oncoprotein is known to be the driver of this rare liver cancer. And usually it's a pediatric cancer. So the idea is this protein, if we can degrade, would be really cool, right? And we know the challenge is we still have the wild type copies, right? So whatever you design to bind and degrade has to be very, very selective for just this and not the wild type copies. That's the case where we have actually designed proteins or mini proteins that bind just the fusion junction. And we know that they are very selective now. And now we are linking them to degrons. So our peptide or mini protein is linked to a degron. So the mini protein goes and binds, recognizes this fusion oncoprotein, and the degron is going to come and degrade this. So we don't have the degradation confirmed yet. We know that our protein with the degron attached can only bind to the fusion and does not bind to the wild type copies. But that's one of the application areas that how we are thinking about it.
Thank you so much for your talk. It was really interesting. I was just curious on how your lab goes about like ensuring chirality in the peptide synthesis.
Yeah. So, I mean, that's a great question, right? So, when we are chemically synthesizing, we are buying sort of like, D amino acid, L amino acids, right? So, really, I mean, at that point, we know what we are coupling. It's really one amino acid coupling at a time. Um, Having said that, there are some rare side reactions where you can see racemization happen, right? And in that case, I mean, really, you would see when you finish it, you run it on HPLC purification, you will see two peaks, and both of them would have the exact same mass, right? So usually in those cases, we just follow the next steps with both the peaks. And ultimately, when we solve the structure, it turns out that one of them had like one amino acid that was flipped in chirality. It's usually a minor reaction. Um, but we trust that because we are including D and L amino acid with known chirality and we know that they are pure enough, that's what we're going to get. But it's kind of one of these difficult problems. There are ways with mass spec, if you go MS to the third, there may be signatures there. Usually when it happens, we just keep going forward with both peaks as separate products. And then ultimately when we solve structure is when we will see it. Yeah.
Great talk. I had a question about the utility of non-canonical amino acids. So do you think they allow you to explore structures that aren't possible with canonical amino acids? Or, if you wanted to design for a certain structure, what would you, if it was possible to do with canonical amino acids, would you prefer to do that? Or is there some added utility to using non-canonical?
Yeah, and that's a good question. So in my head, when I think of non-canonicals, I always kind of like, I have different buckets of them, right? And different buckets have like different functions. For example, D amino acids, right? Just the mirror of that. It actually does help you sample a different space. So like an L amino acid helix versus a D amino acid helix is left-handed versus right-handed, right? So you're actually sampling different structures. Going back to, I think it was Pauling who suggested that if you actually have a L strand and a D strand, the curvature is like flat. Otherwise, you have some slight curvature to it. So you're actually able to sample shapes or conformations that you would not sample otherwise. So that's one. From a drug discovery point of view, most of our proteases have evolved to degrade L amino acids. So if you're making a drug that has to survive the whole gut, having a lot more D amino acids or non-canonicals makes it more protease stable. Right? So there are certain advantages like that. But then we have N-methylated amino acids. And N-methylated amino acids are really good for making things permeable, because otherwise it's very difficult to hide all your backbone hydrogen bond donors. So N-methylated amino acids help there. And then sometimes a lot of these like side chain modifications also help in improving the binding affinity. Now imagine that you have a phenylalanine kind of just sitting, and the pocket is not, there's just enough space that Phe can fit, but maybe a fluorophenylalanine could fit in better, right? So some of these like slight modifications help you improve the binding affinity sometimes by just being, and nature cannot do it, but the non-canonical may be able to do it better kind of thing. So there are different advantages to using them, from stability to improved binding affinity, and definitely more structural space opens up.
Hi, yeah, thanks. Great talk. So when you were looking at these like membrane permeable designs, there's this like confirmation that was more membrane permeable versus not. Does your model output like both of those confirmations for that particular?
So this was, and again, like this was, I mean, as much as everyone and you and we are very excited about the AI methods, physics-based methods had a place. All that work was done with like physics-based methods. And in that case, we would actually generate for every peptide. Like the reason it used to take us like weeks to get to one good design was because we would try to calculate the whole structure energy landscape. So we are calculating all conformations and the energy for each one, calculated energy, right? But those would actually give us designs where we could clearly see that the same sequence can have one low energy minima in this conformation, but same energy, calculated energy, in other conformation as well. So the model was actually giving us these multiple conformations. And I skipped that, but that was one of the most challenging parts of this paper. Like we had 35 X-ray structures in one paper. I would never do that again. Like that was so much work. But how do you show conformational change? So we actually, not just crystal structures, we had to solve 11 more NMR structures in different levels of organic solvents. And we could actually see that in this, as we move to more non-polar solvents, we start seeing more population that is that conformation. And as we go into like more polar solvents, we switch back to the other conformations. So not just in silico, but experimentally too, that we were able to confirm that these conformations actually happen and they do shift as we go from different polarity solvents.
Hi there. Great talk. I had a kind of more general question about engineering membrane permeability. So I'm wondering if there's kind of a general trade-off between, an maximally permeable peptide and one that binds to its target with some specificity, and if in that vein you're exploring maybe using optimal permeable peptides as like a carrier and complexing those with an optimally designed binding peptide.
Yeah, that's a great question, right? So, and that's where kind of like the field is genuinely facing challenges, right? That how do you optimize for multiple properties at the same time, right? It's been a challenge for library-based methods because most of the library-based methods select for target binding. And there have been a lot of companies that have taken high affinity binders and then try to convert them to permeable versions. And that has not worked because once you have optimized for one property so much, it's very hard to kind of like go back to permeability without breaking the binding, right? Our hope is that now we understand permeability, like what kind of shapes are required to make them permeable. And finally, we are good at designing binders. That when we are sampling, we are optimizing for both properties at the same time. Now, having said that, there's still limitations on what kind of targets we can go after, right? If we are using passive permeability, this idea of passively floating across the lipid membrane, that requires some level of hydrophobicity, right? If a peptide is very polar, it's not going to go across the membrane no matter what you do to it, right? So we are selecting our targets to be the targets where we do have a pocket that is decently hydrophobic, right? So that's the limitation. The idea of using them as cargo carriers, in that case, we have to switch from passive, like just floating across the lipid membrane, to more active transport mechanisms. And active transport mechanisms are more, here's a receptor that we know shuttles things. So if we can bind to this receptor, we can take things with the receptor. So we are trying that for a few different, especially for blood-brain barrier. There are a few known receptors that are known to shuttle nutrients across. And if we can make binders to that, then we can take other things across as well. And that's more permissive in terms of what kind of cargo can go across versus passive permeability. So it's again, like, depending on what route you're taking, you have to be careful about what kind of targets on the other side you will be able to hit. And knowing that already limits you. Like some targets, we look at that and clearly it's like, there's no way we can make them bind and be permeable at the same time. So we reject them right away.
Other questions? Because anyone who hasn't asked a question yet? Like we have a great question answering so far. Anything online? I'm not seeing anything online. Well, with that, thank you very much. This was absolutely wonderful. And obviously,
so much.
you
Yeah.
poked a lot of questions, which is wonderful. So people really enjoyed it. So thank you.
Thanks.
Snacks outside and join us for the last week, which will be next week with Mary Claire King. So thank you.